Good morning, everyone. Today we are going to learn about a crucial structure in the flowering plants, the stamens, which is the male reproductive organ of a flower. A typical flower, each stamens has two parts, a long and cylindrical stalk called the filament and a terminal bilobed structure called anther. The filament is attached at the base to the thalamus or sometimes to the petals. If we observe the stamens from 10 different plant species under a dissecting microscope, we will see remarkable diversity in their size, shape, anther attachment. This diversity reflects nature's beauty. Why do you think some stamens are long while others are short? The anther is a tropical angiosperm having bilobed anthers. It means it has two lobes. Each lobe contains two theca, making it dithecous. A longitudinal groove usually appears the two theca of each lobe. In transverse section, the anther looks like a four-sided tetragonal structure. It has four microsporangia at each corner. These microsporangia develops into pollen sacs, which run longitudinally and packed with pollen grains. The bilobed and dithecous nature of the anther is most clearly seen in a cross section. Why is it important for the anther to be bilobed and dithecous? Think about it. Now, if we talk about the structure of microsporangium under the microscope, the microsporangium in TS appears nearly circular. It is surrounded by four wall layers: epidermis, endothecium, middle layers, tapetum. The outer three layers protect the developing pollen and help in anther dehiscence. Dehiscence means to release pollen grains when mature. The tapetum is the innermost layer, each in the cytoplasm and nutrients. Yes, tapetum is a innermost layer and help in nourishment of pollen grains. Why do you think tapetal cells have more than one nucleus? Sporogenous tissue. In the center of the microsporangium, there is a group of compact homogeneous cells called sporogenous cells. As the anther matures, these cells act as microspore. The mother cells undergo meiosis, forming microspore tetrad, a group of four haploid microspores. This process is called as microsporogenesis. What is the ploidy of the cells in a microspore tetrad? Yes, haploids, as they are produced through meiosis. Can you relate the tetrad formation to any other process where four cells are formed from one? Now, the pollen grains formation and its importance. Now let us understand how pollen grains are formed. In the center of each microsporangium, there is a group of homogeneous cells known as sporogenous tissue. Each cell of this tissue is capable of forming microspore tetrad. Thus, each is a potential pollen, or we can say microspore mother cell. The process by which microspores are formed from a pollen mother cell through meiosis is called as microsporogenesis. As a result, each PMC undergoes meiosis, form a tetrad of 4 haploid microspores. These microspores at first remain clustered together in tetrad. As the anther matures and they dehydrates, microspore dissociates from one another and develops into individual pollen grains. Inside each microsporangium, thousands of pollen grains are formed, which are later released when the anther descends. Each pollen, or we can say that each microspore in a tetrad eventually becomes a pollen grain, which is a male gametophyte. A mature pollen grain contains vegetative cell and generative cell. The vegetative cell large help pollen tube growth. The generative cell smaller forms 2 male gametes. Pollen grains are released when the anther descends and they are carried by wind, water, or by insect or animals for pollination. The entire process from sporogenous tissue to formation of mature pollen grains ensure that the male gametes reach the female ovule for fertilization. How does the structure of anther and microsporangium support successful reproduction in plants? Answer it. Now here today we discussed about the structure of stamens, microsporangium, and about the pollen grains. Thank you all.